Okay, little heads up about lab exam tomorrow. If you could come in on kind of the same schedule that you came in last lab exam, that would be helpful. We had a pretty good mix-up of people at different times and things didn't get particularly overcrowded. Format will be the same. When you walk in, over on the side table there will be an answer sheet for you. There will be then questions that will be set up in the same fashion as they were last time at these tables. And then there are, I think, 7 questions on a separate sheet of paper that you'll come and get from me that answers some more theoretical kind of stuff, and you can go next door and do those. Remember that in terms of preparation, that the questions at the end of each lab are very instructive. Some of those you will see, I can tell you that. And then you all got the study guide that I sent out, correct? For break? Okay. So this one is more interpretation as opposed to being quite as hands-on as the last one was. So you're going to have to be able to look at results and determine what those results tell you about the metabolism of the organism. And you do have to know something about some of the chemistry of the reactions. You can use the questions at the end of the labs as guidance for kind of what you need to know. Any questions about lab exam? Okay, I'm going to have to close up the lab at 1 this afternoon so I can start getting set up. What I will do is I will put the EnteroTube scorebook and a couple of computers for ID Bacter over in the principal's lab and Dr. Morton McComb's lab, and that will be available. I'll put a note on the door asking that to be left open this evening if anybody needs to do any last minute things. But this lab will be locked up at about 1. Okay? All right, so we're in the midst of still doing background, so we can kind of start putting the third line of defense together, ultimately. And we're in the process now of talking about antibody molecules. Remember, that is the B lymphocyte, once it's received the proper activation signals, that is going to be responsible for producing antibodies. Specifically, the plasma cell is the one that actually produces the antibody. The 2 signals that typically are required to activate a B lymphocyte to start producing antibody are number 1, binding the antigen, and then number 2, usually but not always, some type of cytokine signal that is secreted by another immune cell. So, here is a representation of the product of those cells becoming activated and then secreting antibody. And here's what the antibody molecule looks like. Should look very familiar. This is the basically same structure as the B cell receptor. Now, not all antibody molecules look like this. In a minute, we're going to talk about the different classes of antibody molecules, and you'll see that this is kind of a general theme of structure, but each class has kind of a variation on the theme. But the idea is the same that we talked about in terms of the B cell receptor. In this particular class, which is IgG, mirror image molecules, 2 heavy chains, 2 light chains. Both the heavy and the light chains consist of variable regions and constant regions. Notice that the constant region of the heavy chains is larger than the constant region of the light chains. These are encoded in the DNA by those gene modules. We have the variable genes. As I said last time, they don't show the diversity and the joining genes in this particular representation, but then these would be encoded for by constant genes. Functionally, there are a couple of parts to this molecule, and one is the antigen binding sites. Those are in this portion of the antibody called the FAB region, specifically at the end of the FAB region. This antigen binding site is identical to this one, so this can bind 2 molecules of identical antigen. Therefore, it's said to have a valence of 2. This region down here is the FC region, And this is the effector, E-F-F-E-C-T-O-R, the effector region of the antibody. You can have hundreds of millions of antibodies that belong to the G class. What does each one of those differ? In this region right up here, in the antigen binding site. But if you are a member of the G class, then this region of the antibody structure will be the same for every one of those IgG antibodies. So this is a kind of a generalized part of the structure of all Gs. And then up here, it varies from different IgG to IgG depending upon which particular type of antibody it is. The effector region gives a generalized function to that class of antibody. And we talked last time about what some of those generalized functions were. Some antibodies can cross the placenta, others can't, to give maternal protection to the fetus. Some can be secreted, others can't. Some can fix complement, others can't. And so that is determined by this particular FC region down here. So this defines what class the antibody belongs to. Another thing we're going to see is that once an antibody is produced by a plasma cell, if it gets the right signal, the plasma cell can switch over and start producing a different class of antibodies sometimes. So it might begin producing an antibody class called IgM. But then if it gets the right signal, it can start producing IgG, or it can start producing IgA. That particular phenomena is called class switching, and we'll take a look at how that happens in just a second. But before we do that, let's get into this. We've alluded to this a little bit already. What's the benefit of producing an antibody? Why is it protective? Why do you have this elaborate system of activation of B lymphocytes in the plasma cells to secrete these protein molecules? What do they do for you? Well, an antibody is going to bind to some foreign antigen with which it's paired. And that in of itself can have several consequences. One consequence is that the antibody can serve as an opsonin. An opsonin is an antibody that enhances phagocytosis. It makes it easier for those neutrophils and macrophages to recognize you if you're coated with antibody molecule, if you've got that kidney sign plastered onto you. What specifically are the macrophages recognizing? Well, okay, over here on the left-hand diagram, you've got these antibody molecules, there's one, there's another, bound to certain antigens on the surface of those bacterial cells, okay? And that little knob sticking out here on the antibody is the FC region. There are receptors on the surface of neutrophils and macrophages that recognize FC antibody regions and bind to them. And because the antibody is bound to the antigen, which is bound to the cell, then you've got this kind of sandwich where now the macrophage or neutrophil is bound to the antibody, which is holding capture the pathogen. So this enhances the possibility that phagocytosis will occur. Another way that antibodies can protect you from infection is that virus have receptors on their surface which have to bind to cell surfaces of your tissue in order for infection to occur. If a virus can't attach, it can't infect because it won't be brought into the cell. So what can these antibodies that are being produced do? They can bind to the surface of the virus and basically block those viral antigens and prevent them from binding to receptors on your cells. Another thing that can happen is that antibodies can cause clumping of pathogens. Remember that I told you that it was significant that antibodies had a valence of at least 2. Some classes of antibodies have a valence more than 2, but you have to be able to bind at least 2 antibodies. Okay? Or 2 antigens, sorry. So look at the consequences of that happening. One antibody molecule can bind to 2 pathogens by its 2 arms. And then another antibody molecule can hook that pathogen to this one. The fact that antibodies can bind more than one antigen allows cross-linking of those antigens. Formation of a clump. And these clumps are more prone to be phagocytized than individual bacterial cells are. The neutrophil or macrophil goes, oh, hey, cool. Here's a big clump of bacteria. I'm going to gobble it all up. This is also, we're going to see later on, the basis of the serological reaction of blood typing. That's one of the ways how you can tell what your blood type is by mixing your cells with particular antibody. And if the antibody reacts with your cell's antigens, clumping will occur, and you can see a visible reaction taking as that clumping occurs. Another really important part of your defense mechanism against infection is the complement system, and particularly the classical complement pathway. And what does that require? That requires initiation of the complement cascade by what? An antibody reacting with a bacterial antigen. So here, because those bacterial cells are now coated with antibody, if those antibodies have an FC region that can fix complement, a complement cascade will form, and holes will be punched in the membrane of that pathogen, and it'll be lysed. Precipitation reactions occur in much the same way that agglutination reactions occur. It's the result of cross-linking of antigen with antibodies. What's the major difference? In agglutination, the antigens are typically part of cell surfaces. In precipitation reactions, the antigens tend to be soluble molecules. It might be a protein toxin, for example. But clumping those soluble toxins together can again make a clump, which then makes it easier for the body to eliminate that particular threat. There's one final mechanism that I want to briefly mention whereby antibodies can protect you, and that's a phenomenon called ADC, antibody-dependent cytotoxicity. This is kind of an interesting phenomena because in this particular case, the antibody actually serves as a catalyst for a reaction. And remember, they are proteins, okay? So they do have potentially catalytic capability. And one of the things these catalysts can activate is the production of reactive oxygen species. And hopefully by now you appreciate the fact that reactive oxygen species are one of the mechanisms that your immune cells can use, particularly the neutrophils, to kill pathogenic microorganisms. So, there's a lot of benefit in producing these particular molecules. They protect you in any number of different ways. Okay, so here are the classes of antibodies that exist. 5 classes of antibodies, each indicated by a letter designation: IgG, IgA, IgM, IgD, and IgE. And these first 3 right here are the ones that we're going to spend most time talking about and comparing. We'll make brief reference to this one. This is basically the same thing as the B cell receptor. Let's start out by looking at IgM first. If you look at IgM, you go, wow, this is a pretty complicated molecule, especially compared to IgG. In reality, IgM is a pentamer, a cluster of 5, of these IgG molecules stuck together. And that's reflected in the fact that it has a much larger molecular weight. There is a polypeptide chain called the J region, which joins these IgG molecules together to form this big pentameric molecule. What's the valence of IgM? 10, because it's got 2, 4, 6, 8, 10 identical antigen binding sites. If we look at the total percentage of antibody that's circulating around in your blood serum, this is a relatively small portion of that total circulating antibody. And it doesn't last very long. It only lasts a few days in duration. What are its effector functions? What are some of the things it can do? Well, one of the big things it can do is it can fix complement. Okay? But it does not have the ability to cross the placenta. Why do you suppose not? Why do you suppose it can't cross the placenta? Too big, yeah. Way too big. It doesn't have any particular affinity for other cell types. And in a minute you're going to see that some of these other antibodies have a particular affinity for other cell types. That Fc region binds to receptors on other cell types in your body. So what does this molecule do? Basically, this is the antibody that is first produced when B cells become activated and start cranking out antibody. So IgM is the first class of antibody produced. Once a B lymphocyte starts producing IgM, if it gets the proper signals, that B lymphocyte can switch over and start producing IgG or possibly IgA as well. The most common class switch that occurs is IgM switching over to IgG. So what does that class switching involve? It involves another genetic rearrangement in the genome of those lymphocytes. I know this is really hard to see over here. You'll have to look at this on your printout or on your computer. But here are the battery of genes that are responsible for encoding antibody structure. And we've already had a genetic rearrangement of the B, the D, and the J components. But now we need to stick that together with the C component to form a heavy antibody chain or a light antibody chain. In green here are the various C modules. There's one for IgM, one for IgD, G, etc. Okay? So the genes get rearranged, and originally it is the M component that is being paired with the DDJ, and that's what's forming your heavy chain or your light chain. So therefore you produce an M-class antibody. But if you get the proper signals, cytokines, okay, genetic rearrangement changes a little bit, and the genome starts getting spliced differently, and instead we now associate a class G module with the BDJ, and now therefore we're switching over and producing a class G antibody where we were previously producing the class M antibody.
I might be oversimplifying this, but so is it just basically the constant region that's changing in this?
Yep.
Okay.
Yep. Yeah, because remember that we're still producing an antibody with the affinity for the same antigen. And that is a function of that variable region right there. So we're not rearranging that. We're just pairing it with a new constant region. So now it can do something different as a class of antibody. Okay, previously it couldn't cross the placenta. Now it can. Okay? So, how does IgG compare to IgM? Well, it's smaller. It's only a dimer instead of a pentamer. So it's got a valence of 2 instead of 10. Well, look at this number here. This is pretty significant. It's got by far the greatest percentage of all antibody classes of antibody that's circulating around in your blood serum. So if you get an antibody test for something, we'll talk about this test later on, then likely speaking what you're doing is you're analyzing your IgG antibody populations. Because that IgG makes up about 80% of the circulating antibody. Notice that it's got a much longer average lifetime than IgM does. Average life of those antibodies is about 23 days. Okay? A month, approximately, before it's degraded. Notice, too, that it's got a lot of biological function. It crosses the placenta. It fixes complement. It also neutralizes some toxins and some virus. This also has an affinity for phagocytic cells. So that Fc region of the IgG is recognized by phagocytic cells. So IgG is an opsonate. It coats the pathogen and makes it more likely to be phagocytized by neutrophils and macrophages. IgG tends to give you long-term immunity. Sometimes it's referred to as the memory immunity. But it's really important to understand that the only way you can produce IgG is that that lymphocyte gets the proper cytokine signals to switch over from producing IgM to IgG. And so it takes an additional stimulus for this class of antibody to be produced. IgA. IgA occurs in two forms. It occurs in the serum form and the secretory form. And obviously the secretory form is the form that's going to be secreted by various glands of the body. Breast tissue can secrete IgA into breast milk. Epithelial cells can secrete IgA onto their mucosal surfaces. So your gut epithelial cells, when lymphocytes become associated with those, can start secreting out IgA antibody, which can coat the surface of your gut or your nasal passages, for example. Why is that significant? Because those are common portals of entry for pathogens. And so if that antibody is sitting there waiting for the pathogens, that gives you an enhanced level of protection. It's actually the tissue that secretes the antibody. So the breast tissue secretes the IgA. Now the IgA is being produced by lymphocytes. First it's going into the serum, and then it's being gathered together basically, and then secreted by the appropriate tissue, by the epithelial tissue or the breast tissue. And there's a little polypeptide chain called the secretory component, which helps to facilitate that particular secretion. So not a lot of this circulating around in the blood. Why? Because most of it's being gathered up and pushed out of the body and being secreted onto cell surfaces or into tissue fluids like breast milk. Relatively short lifetime, but notice that it has an affinity for epithelial cells particularly, and so this is our major secreted antibody. So now this gives us a level of protection not only in the fluids of the body, but on cell surfaces in some of the vulnerable areas. IgE is the antibody of allergy. IgE looks a lot like IgG. It's a dimer as well. It does play a role in protection against parasitic worm infections. But it is also developed into the antibody of allergy. What happens in allergic reactions is that people who suffer from allergies overproduce IgE antibody in response to things that shouldn't be that threatening, like pollen. You have some genetic predisposition to produce more IgE than normal people do. And what happens? This IgE antibody has a particular affinity for mast cells and basophils. Mast cells are basically cousins to basophils. They're found in connective tissue. Both of them are granulocytes, and their cytoplasmic granules are filled with substances that stimulate inflammation. If enough IgE antibodies are produced and start binding to mast cells and basophils, those cells can become sensitized. They become fragile, basically. And then the next time an antibody, IgE, is produced in response to allergen, the sensitized cells bust open. They degranulate. And they start releasing all their inflammatory substances. And now you have allergy. So can you suffer an allergic response the very first time that you're exposed to an allergen? No. It takes time to develop enough IgE to sensitize your cells. But then once they're sensitized, when you're exposed to the allergen again and you make more IgE, those sensitized cells degranulate and you've got allergy problems. So if you've had bee stings, for example, and you're becoming increasingly responsive to a bee sting, every time the response gets worse and worse, you're getting sensitized to that particular allergen. And you need to be careful, because once you're sensitized, if you get another bee sting, that induces IgE, and those sensitized cells start to degranulate. And if too many of them degranulate, you can go into anaphylactic shock. And if you're not carrying around an EpiPen or something like that, you can die from that anaphylactic shock. So this is kind of a perversion of the immune response. This is an immunopathology. This is an immune response gone wrong. The normal intention of this particular class of antibody is to protect you from those parasitic worm infections. But you know, in developed countries particularly, we don't have that many parasitic worm infections anymore, in part because we have good sanitation systems. And what seems to have happened is that as that threat has been eliminated, that the response of these antibodies to allergens has become more pronounced. And so we've seen an increased amount in allergies over time. It's kind of an interesting interplay. Okay, this slide looks a little complicated. It's not really as bad as it appears. But this gives you a good summary of kind of what's going on as you're exposed to allergen the first time and then subsequent exposures. The first time that you're exposed to an antigen, the body generates what we call a primary immune response. So that's to the left of this particular diagram up here. So if you'll notice, whoops, on this particular graph, we're plotting antibody titer, the amount of antibody produced, versus time. So here's your first exposure to the antigen. That generates a primary response. What's going on? That antigen is stimulating educated but dormant, naive B cells that are sitting there waiting to be activated. And that's going to take some time. And so one of the things that characterizes this primary response is this period of latency. It can be several days before those B cells get properly activated. And what are they going to start producing? The plasma cells will start producing IgM. So we start seeing a rise in the first class of antibody produced, IgM. But IgM is a relatively short lifetime. The other thing that tends to happen during this primary response is that in addition to forming plasma cells, we form memory cells. And memory cells have undergone class switching. So once they are stimulated by the antigen, they start producing the IgG class of antibody. So you see the total antibody response is indicated here in purple, and it is made up of first IgM production, later followed by IgG production. Okay? So this is already a memory response within this primary part of the exposure. Eventually, if the antigen is eliminated and the threat is over, then these cells will shut down and antibody production will cease and levels will eventually fall. But we still will have these memory cells sitting around there, populating your lymph nodes, populating your spleen. What if we get a second exposure? Well, now we're into a secondary response. Secondary responses are also called anamnestic responses. This is a memory response. So what do we see? We see a dramatic increase in antibody production from these memory cells almost immediately. Notice that the latency period isn't there. Why? Because these memory cells, which were already present, can be activated to produce IgG almost immediately. They've gone through their class switching. They were sitting there dormant, but now they get the proper signals, and now they start doing clonal expansion and start really cranking out IgG antibody in large amounts. Now, sitting in your lymph nodes will be some naive lymphocytes that didn't see the antigen the first time in the primary response. So they're going to be activated. And what are they going to do? They're going to produce IgM. But it's going to take a while. There's still that latency there. But this is where you get your protection from this memory response. So if we generate a primary response by immunization, and you trick your immune system into thinking, hey, I've seen that before. I now have memory cells that remember that antigen. Then if you're exposed to the real antigen, the pathogen or the toxin, you can respond very, very quickly and get high levels of protection almost immediately. But if the first time you see the pathogen or the toxin is a primary response, you might not make it through that latency period while your immune system is building up enough antibodies to protect you and allow you to survive. Now, does this memory last forever? No, not necessarily. Generally speaking, you get a better memory response if you are exposed to the real thing as opposed to some weakened version of the real thing that you get in an immunization. So that is why many immunizations require boosters, kind of reintroducing yourself to the fake antigen. So every 10 years you should get your Tdap, your tetanus, diphtheria, pertussis, to kind of re-educate your immune system. But if you've gotten measles as a kid, you're protected for life. You've seen that real thing and it's giving you a more robust, strong immune response. So what this slide illustrates is the different types of immunity that you can get and develop over your lifetime. This would be acquired immunity. This would be third line of defense immunities due to production of antibody. Acquired immunity is broken down into two categories. There is natural acquired immunity and there is artificial immunity. Each of those two subclasses are subdivided into subclasses themselves. And those particular subclasses are active and passive immunity. So there can be active natural, there can be passive natural, there can be active artificial, and there can be passive artificial. What's going on in each of those particular cases? Okay? The active natural immunity occurs when you're exposed to the real thing, a pathogen or a toxin. That would be a primary response. Hopefully you will survive that primary response and now have memory. Hopefully the disease will be self-limiting and you'll survive. Passive natural immunity is the result of getting antibodies from your mother, either by crossing the placenta, so the IgG she's producing in response to what she's encountering in terms of pathogens and toxins can be transferred to you across the placenta, or IgA antibodies she produces can be produced in breast milk. Here, though, you're getting preformed antibodies. And remember that antibodies have a limited lifetime, and so they don't last forever. So this gives you temporary protection. This gives you long-lasting, maybe lifetime protection if you survive it. Artificial immunity is induced by a medical procedure. You're not seeing the real thing. You're seeing some variant that's been created, which is intended to trick your immune response. Active artificial immunity is immunization. And again, the concept is we want to shift you from this status to this status over here by immunizing you. So you eliminate that latency period when you're really exposed to the real thing. Artificial passive immunity involves getting antibodies not from your mother, but from another individual. One way in which you can get antibodies from another individual is to receive a shot of pooled gamma globulin. Pooled gamma globulin is obtained from blood donors. It's said to be pooled because the blood serum from several different donors is pooled together. Why? Because the more individuals you include in this pooled sample that you're injecting, the more protective antibodies there likely will be, the more different kind of protective antibodies. So if I just gave a gamma globulin shot from Brandon to Richie, he might have certain protective antibodies in his syrup, but not others. But if I pooled the results of Sarah's and Jake's and Brandon's and Ethan's together and got this pooled gamma globulin fraction, just because they have different exposure histories, they're probably going to have more variety of antibodies present in that gamma globulin that's given. Okay? But again, these don't last forever. Those antibodies don't last forever. So it gives you fast but temporary protection. You don't have to wait for that latency period because you're not producing your own antibodies. You're giving people antibodies from other individuals that have produced them. Pooled gamma globulin shots are sometimes given when there is a foodborne hepatitis outbreak. And all of a sudden they decide that, wow, several people visiting this particular restaurant are coming down with hepatitis. So they put out a public health alert and say, if you've eaten at Steak and Shake in the last 30 days, you should go to your doctor and get a test for hepatitis. And in the meantime, your doctor might say, it's going to take a while to get these results. I'm going to give you a pooled gamma globulin shot to protect you, hopefully, against this disease while we're waiting to get the results and put together a treatment plan. Okay, now we've got the background that we need to put everything together. We know about receptors. We know about MHC receptors. We know a little bit about CD receptors. We know about B and T cell receptors. We know about antigens and what they are. We know about antibodies and how they protect you. We know a little bit about some of the cytokines that do cell signaling. So the last thing we're going to do is talk about this adaptive immune system now, which is made up of two overlapping portions or arms. We call those two overlapping portions cell-mediated immunity and humoral immunity. We've already mentioned the differences between these two types of immunity. Cell-mediated immunity tends to be directed toward your own cells and destroying your own cells that have gone bad. They become cancerous or they become infected by virus. They need to be eliminated. Cell-mediated immunity is primarily a T cell process. T killer cells are involved. T helper cells are involved. And there also is some type of antigen presenting cell involved. So CMI involves T killers or T cytotoxic, same thing. A class of T helper cells called T helper 1 cells, and some type of an antigen presenting cell. It could be a macrophage, or it could be a dendritic cell. But the goal of this cell-mediated immunity is to lyse host cells that have somehow become infected or damaged. So basically what we need to do to put this together is to talk about some of the interaction that takes place between these players and see what some of the receptor interactions are to make sure this happens properly, and also see what some of the cytokines involved are. Humoral immunity also involves the reaction of antigen-presenting cells. And they could be macrophages, they could be dendritic cells. B lymphocytes. And a different type of T helper cell called T helper 2 cells. When the right receptors match up with the right antigens or the right cytokines are produced, what happens? What's the goal of this particular response? Antibody production. And all of the benefits associated with producing those particular antibodies. So we're going to do this on Thursday. I would recommend that if you can find time, that you look at the slides, the last few slides of these, and particularly this group of slides right here that shows some of the interactions. And just start looking at them and start reading through them and see if you can start making any sense out of them at all. Because this is the kind of information that the first time you look at it, you go, man, I'll never get this wrapped around my head. But as you go through it a little bit, and then I can talk you through part of it, and you go back through it again, this is the kind of thing that you would really benefit from some pre-class preparation and not walking in cold. Because there's a lot of interactions that take place and a lot of different things to keep straight. Okay?
ID factor.